All right, Dr. McNeil talking to you about DNA replication. Okay, so what's the deal? To replicate either linear or circular chromosomes, say if you have a bacteria, you have a double-stranded piece of DNA. That's a circular chromosome. There's a double-stranded DNA. Is no different than the linear chromosomes in eukaryotes, complex animals. And the first thing that you need to undergo DNA replication is to open the DNA up. And this happens at a given place on both the bacterial chromosome, called the origin of replication. And since this origin of replication is on the circular chromosome, one origin is going to be enough to replicate DNA moving that way and moving this way, and it will replicate the entire chromosome. In eukaryotes, that's not true, and you need many origins of replications throughout a linear chromosome so that we can write DNA in both directions from all origins in order to replicate our entire chromosome. Okay. There's two reasons for this. One is that bacterial chromosomes are generally about 4 million base pairs. But the human genome has 3.3 billion base pairs. So what we really have here is a need for lots of origins to fire, lots of origins to turn on DNA replication so that we can replicate our humongous genome at a reasonable speed. And bacteria don't need that because they only have 1 million bases. Okay? So what I'm going to do is teach you about the bacterial origin of replication. And it's so similar to ours, and the replication bubble that this will form. It's so similar to ours that we're not going to worry about comparing them. I'm just going to tell you how it works. Okay? Here we go. So what's the very first thing that you need to happen? Well, you have your double-stranded DNA, and you have to open it up. So here's double-stranded DNA. Is it open? And coming back close. So if this is bacteria, remember this ultimately 4 million base pairs away will close the circle. But we aren't going to worry about that. Then we need to assign polarity to each of our DNA molecules, because all of our DNA molecules run in the 5' to 3' direction. So I'm going to say 3', 5', got to follow it, 3, 5', and 3'. So we have anti-parallel double helix of DNA. Okay? And one key about DNA replication is DNA polymerase writes new DNA in a 5' to 3' direction using a 3' to 5' DNA template. Okay? So because of this, because of the use of the DNA template, we say that DNA polymerase is a DNA dependent DNA polymerase. Why? Because without a DNA template, it can't do any work. So it's DNA dependent DNA polymerase. The other thing that you should know by now is what I always say about DNA polymerase. You'll know this from lab, from doing PCR, is that DNA polymerases cannot start new DNA chains. It cannot start new DNA chains. It can only extend free 3' OH groups. Okay, so we feel good about that. We're going to move on. So what are we going to do? Well, we need a 3' OH to direct where our DNA polymerase is going to go. Okay, so we're going to have an enzyme that lays that down, and that enzyme is called primase. Okay, and primase needs to use a template in the 3' to 5' direction. So if we're going to write new DNA in the 5' to 3' direction, primase would lay down a 5' to 3' OH template. But primase is an enzyme, right? It's not a DNA polymerase. It can't be, because DNA polymerase cannot start new chains. So primase is a DNA-dependent RNA polymerase. It writes short strands of RNA complementary to DNA at the origin of replication. Okay? So that's our first basic step for replication. Now, this will allow this side of the replication bubble to move to the left. A similar process, which I'm only going to diagram the first primer for that, starts up here. We have a 5' to 3' RNA primer here as well. And that will lead to the replication fork now, moving that way. So this whole thing is a replication bubble, but this side and this side are replication forks. Okay? So we now have the basis for what we need to do in replicating our DNA. But what don't we have? Well, we don't have any protection, really, when you think about it, on this DNA. So normally our DNA exists as a double-stranded chromosome, right? But all this is single-stranded DNA just hanging out. And that is not optimal. The reason it's not optimal is that all this single-stranded DNA looks like a viral genome, and it's eligible to be torn up by DNA-modifying enzymes. So, a special set of proteins, which I always draw as 4 dots, don't ask me why, called SSBPs. That's single-stranded binding proteins. Both bind to single-stranded DNA, and because this is a protein wrapping this DNA molecule, it's protecting that DNA molecule from being degraded. Okay? So now we've got protected DNA, we have an open origin of replication, and we're ready to write DNA. Okay? But we have a lot more going on than that. So if you think about it, if I were to roll up a rubber band, and then I were to push all the coils to one side, it gets tighter and tighter and tighter. So that's essentially what's going on right here. And there's a few things that are working to both open this helix up, but then also relieve tension. Okay? So I'm going to draw these proteins as shapes, despite the fact that they're clearly a protein. So what do we have? We have a protein, helicase. Right? It's an enzyme, ASE, helicase. And that is an enzyme that destroys helices. So that makes double-stranded DNA into single-stranded DNA by breaking the hydrogen bonds in between the two strands. More than that, so as helicase goes down and it's breaking the double-strand bonds, this DNA just slightly upstream of it is getting tighter and tighter and tighter and tighter and tighter coiled, right? But you can't have that, because if you like torque on a rubber band too much, eventually they'll break. So what you have is another enzyme that goes just ahead of the DNA replication fork called topoisomerase. Some people call this, it's actually technically topoisomerase 2, and some people call this DNA gyrase. And what's topoisomerase going to do for us? It nicks a single strand of this DNA and allows it to unwind itself to relieve tension. So this. Relieves tension at the replication fork. Right? So this isn't only happening on one side. All of these things are happening analogously over here as well.